So we will discuss the paper 2 first for your trial exam. So question 1 shows you a electron micrograph, transmission electron micrograph, and they label A as well as mitochondrion. They've given you the magnification. So the question asks the student to state how it is possible to deduce that figure 1.1 is a transmission electron micrograph and not a scanning electron micrograph. So it's a TEM and it's not a SEM. So this is talking about electron micrograph, and you know that you have learned two main types: transmission electron micrograph. So for light microscope, we shine light across the object, correct? So for electron microscope, instead of light, what do we use? Cassandra? What do we use? Cassandra? Light microscope, we use light. Electron microscope, what do we use? Cassandra? Cassandra? Okay, Cassandra doesn't seem to answer the question. Okay, Angela? Please respond so that we can move on faster. This discussion is not only I giving you answer. You need to participate to show that you actually understand. Angela? Angela also not responding. So I assume that they do not know or they are not here. Cheng Han sure know the answer.
Electrons.
Electrons. So electron microscope will actually shoot the electron towards the object, right? So you shoot the electrons towards the object, and then when it comes to the object, there are certain part of object that's electron dense. So with the more atoms that you have, right? You got a lot of electrons surrounding the atoms. So the more electrons that will actually repel this, the more electrons you have here to repel the electrons. So electron cannot pass through. So electron only pass through the region which is having less molecule or less atom. That's why you can see this diagram here. No, the parts which is dark in color is because the electrons cannot pass through. So when it cannot pass through, when it comes to the screen will have got less electrons, so it becomes dark color. Let's say this part here will be dark color because electron cannot pass through. This part will be bright because electrons can pass through. Right? The electrons can pass through, that's why it's bright. So this is the principle of electron microscope. So when you study biology, everything they've taught you, you need to learn up. Okay, so then you got TEM and SEM. TEM, transmission, means to pass through. Whereas SEM is a scanning electron microscope. So we only use the electron to be reflected from the surface of the object because we have stained the surface of an object with probably gold or something that will reflect the electrons. So therefore, it gives you a surface view or surface contour of the object. So this will give you a 3D view of the object, in this case, the cell. So you only see the surface view because we have stained the surface with gold. Okay, and therefore it reflects the gold. So the electrons cannot pass through the cell. So for TEM, the electron can pass through the cell, so you can see the internal structure of the cell. Example, the mitochondrion, you can see here, Golgi apparatus, 70S ribosomes are the structure you can see, so you need to mention those structures here. Okay, and it's 2D, so that's the answer. Okay. So next, structure A is part of the network of membranes inside the cell. So I already said it's a network of membranes. So students who write network of membranes, I should not give any mark. They have given you the mark, please minus 1 mark away. Okay. It is the site of modification of protein. So they even say that this part modifies the protein. So straight away, we know that this was the Golgi apparatus, right? Such as glycosylation. So the answer is Golgi. Golgi must be spelled as a capital G. Strictly speaking, if you use small letter G, in exam, you will not get any mark because capital letter. Golgi apparatus. There are some students who still use small letter. Please, when you learn biology, you need to pay attention to the spelling of the word, whether you start with capital or small letter, whether you should underline, you do not underline, singular, plural, all the spelling is you need to know. Okay, you cannot simply give. Then, of course, you can use Golgi complex, Golgi body also can. Part 2 state one structural feature of A as shown that supports your answer in B. Of course, you said network of membranes, I will not give you mark. Remember, membrane-bound sets, we have learned to give a name to membrane-bound sets. What is the name given? EJ, give me the name of membrane-bound sets.
Oh, no. Is it?
Yes? Asta. What is the name of membrane-bound sets? EJ? You don't know, is it? Okay, EJ don't know. So Joey?
Hello?
Cisterne. Okay. Simba says cisterne, very good. Cisterne. Cisterne is the name given to membrane-bound sacs. So Golgi apparatus is made up of stacks of cisterne. This is the first thing, stack of cisterne. They are not interconnected. So some of you who write this is rough ER. Rough ER, the cisterne are interconnected one, but this one is not interconnected at all. So it's stacks of cisterne that are not interconnected. And not only that, another thing is it is not connected to the outer membrane of nucleus. Okay? If it's rough ER, is actually not only interconnected, it's also connected to the outer membrane of nucleus, but this one is not. And lastly, at the edge of the cisterne, there are vesicles, okay, secretory vesicles or vesicles that butt off the edge of the cisterna. So this one you can see from the diagram here. See this one here, this one vesicle, this one is enlarged. So this one already a complete circle, so it's already a vesicle that butt off from the edge of the cisterna. But this one here is enlarged, but which has not butt off, same as this part here is enlarged. So you can either write there having cisterna at the edge or the edge of cisterna is enlarged. So that's the next one you can write. Edge of cisternae enlarged. So you need to know that cisterna is for one. If you write cisternae, that means there are a few, then you must make sure that the diagram actually shows a few of them having that. So this one, okay, you got one here, you got two here. So if you write cisternae, it'll be correct. If there's only one in the diagram, you write cisternae, you will not get the mark because it's wrong. So you have to be very careful with spelling for singular and plural when you describe your answer. Must be number one, accurate. Number two, crystal clear. Okay. So part C is you need to calculate the actual diameter XY. And not only that, you need to show your answer in the nearest whole number and it must be in nanometers. So majority of students lose mark here, as in you don't score the full marks because you do not know how to convert from cm or mm to nanometer. Number two, you did not show the formula. So this is where majority of students lose the mark. Okay. Now they already said very clearly that write down the formula that you will use. So therefore you need to write down the formula. Now the formula that you write down, you cannot just write, frankly speaking, you cannot just write m is equals to I over A, because I taught you that if you want to use a symbol to represent something, you need to tell me what this symbol means. So this is magnification. So you have to write here like this. I is equals to image size. And then A is equals to actual size. You need to spell out fully when you use symbol, because these are not official symbol. Okay, not official symbol like in physics, the official symbol for mass is M, force is F, right? Acceleration is A, gravitational force is G. These are all called official symbol, but this is not. So you have to spell out fully. So therefore, if I've given you mark, just tell yourself that in actual exam, you may not get a mark. Okay, if you've written M equals I over A. It's a 50-50, it's up to the examiner to decide to give mark or not. So you want to write answer that you 100% will know that you get the mark, not hope that the examiner will give you mark. Okay. So write answer that will give you 100% the mark. Then secondly, what is the answer here? Second mark is given for correct calculation all the way until nanometers. Okay. So let's look at the mark scheme. So this is the answer here. So first mark is given for the formula that's given. Second mark for calculation. So when you measure the X and Y using your ruler, now you already know in the diagram that the magnification is given. Okay, so magnification is given here. So M is already given. And then you measure using your ruler from X to Y, that will be your I. Then you can calculate A. Okay, so the I from the diagram is actually 14 mm. So we allow a range of answer from 14 plus minus 1 mm. So that means to say that your measurement can be 13, 13.5, 14, 14.5, and 15. So we accept only these measurements. So based on these measurements, after you calculate, the actual length should be this one only and nothing else. So based on the value that you measure, these are the only possible answers in nanometers. No other answer acceptable. Now, let's say, for example, teacher, my calculation is correct. Sorry, there are some students' calculation correct, but the rounding up part was wrong. Let's say, for example, 276.66667, for example. The student round up to become 276. Of course, this is wrong. So how to give mark when your round up is wrong? Everything correct, then the round up wrong. 276.6 supposed to be 277, right? The student will write 276. So therefore, wrong. Okay?